Mutations in TUBB8 and human oocyte meiotic arrest. Definition: human reproduction depends on the fission of a mature oocyte with a sperm cell to form a fertilized egg. The genetic events that led to the arrest of human oocyte maturation are unknown.
I'm Fu Tang from Peking University. There are more than 200 types of cells within a human body. Probably one of the most important types of them is human eggs, because they are the only type of cells that can transmit all the genes from a mother to her children and keep the human species immortal. The human eggs are also very special in the way that every egg is genetically different. In fact, this is why every one of us is genetically unique, except identical twins. Every egg cell is genetically different, so you have to analyze them one by one to really understand their unique genetics. We recently published a paper in Cell to show that by using cutting-edge technology of mobile single-cell genome amplification and deep sequencing, we can get a better understanding of these mysterious human eggs. Firstly, in a human egg, the two sets of genes coming from grandmother and grandfather need to be rearranged and mixed properly, similar to shuffle of pokers. Every of the gene strings called chromosomes need to be exchanged with its sister chromosome. The rearrangement in eggs is more thorough than that in sperm. We now have a better idea why that happens. Secondly, for the first time, we freeze a woman's genome by sequencing her eggs. There are two sets of genes in every cell of a person. It's difficult to separate them to two distinct groups accurately. Now, taking the advantage that within a byproduct cell of an egg, called a second pole body, there is only one set, but not two sets of genes. We can easily separate the two sets of genes of a woman into two distinct groups. In this way, we can get a better understanding of the genetics of the woman.
I'm Dr. Qiao from Peking University Third Hospital. In vitro fertilization is a powerful technology to help infertile couples to get their own babies. The first IVF baby in the world was born in 1978 in the UK, and the first one in China was born in 1988 in our hospital. Although the IVF technology has been successfully used for more than 35 years, the success rate is still relatively lower, around 30 to 40%. One of the main problems comes from genetic anomaly of human eggs called aneuploidy. It has been shown that when a woman is older than 35 years old, more than 30% of her eggs are genetically abnormal as aneuploidy. These abnormal eggs will lead to either miscarriage or birth of the baby with genetic disease. The real problem is that there is no accurate, efficient, and lower-cost way to detect them. Because of the genetic material within an egg is so tiny that it's too difficult to detect this abnormality with standard molecular biology tools. Fish, SNP, and CGH array have been developed successfully but with limited resolution and accuracy. We analyzed the two pole bodies of one human egg by deep sequencing, simply by counting the number of chromosomes in this byproduct cell as 0, 1, 2, 3, 4. We will know that if the egg is a normal one with two complete sets of genes, or an abnormal one lost of again some copies of genes. More than this, we can also screen gene mutation in these eggs inherited from the mother. If the mother has some gene copies out of work, we will know that if their out-of-work gene copies are transmitted into these eggs or not. In this way, we kill two birds with one stone. One deep sequencing analysis to avoid two types of genetic problems.
Application in medicine. It was sequenced the exons of 5 members of a 4-generation family, 3 of whom had infertility due to oocyte meiosis arrest. It was performed Sanger sequencing of a candidate gene, TUBB8, in the DNA samples from these members, additional family members, and members of 23 other affected families. The expression of TUBB8 and all other tubulin isotypes was assessed in human oocytes, early embryos, sperm cells, and several somatic tissues by means of a quantitative reverse transcriptase polymerase chain reaction assay. Pros and cons. Pros. It was evaluated the effect of the TUBB8 mutations on the assembly of the heterodimer consisting of one A-tubulin polypeptide and one B-tubulin polypeptide in vitro, on microtubule architecture, on microtubule dynamics in just cells, and on spindle assembly in mouse and human oocytes. Cons. We identified 7 mutations in the primate-specific gene, TUBB8, that were responsible for oocyte meiosis. The mutations affect chaperone-dependent folding, cause catastrophic spindle assembly defects, and maturation, arrest, and expression in mouse and human oocytes.
Time-lapse monitoring of zona pellucida free embryos obtained through IVF, a retrospective case series. The zona pellucida is a thick extracellular elastic coat surrounding the oocyte, composed of a 3-dimensional structure of interconnected sulfated glycoproteins. It has multiple in vivo functions, which among others include protecting the oocyte developing embryo, selecting a single fertilizing spermatozoon, and providing mechanical support by preventing blastomer separation before the compaction stage occurs. Previous case reports have already described that mature zona-free human oocytes can be successfully fertilized through ICSI and could develop further to the blastocyst stage. However, so far only one report described the successful delivery of a healthy male newborn following fertilization of a zona-free egg, blastocyst culture, and elective vitrification. This retrospective case series included all 32 patients between October 2012 and June 2014, where an oocyte with a damaged zona pellucida was retrieved and was afterwards removed from its envelope. Patients have undergone natural cycle IVF or minimal ovarian stimulation exclusively. Resulting embryos were cultured until the blastocyst stage and were electively vitrified for subsequent use. Baseline characteristics of the included 32 study patients are summarized here. The majority were 40 years or older, had primary infertility, and were nulliparous. Bologna poor responders were overrepresented, and most patients have already undergone IVF treatments elsewhere.
Issue applied in Ecuador. In Ecuador, there is not too much genetic studies Both doctors and scientists stated that an important event of oogenesis is vitellogenic period of nuclear and cytoplasmic growth that occurs after the first meiotic arrest independent of prophase 1, in which vitellogenic protein is synthesized in the liver and carried by the bloodstream to the ovary. Within the oocyte, this protein is converted into lipobictina and phosphitine, proteins that are part of Golgi platters, needed for subsequent embryo nutrition.
Meiosis consists of 2 consecutive nuclear divisions per round of DNA replication. The two, called meiosis I and meiosis II, result in 4 haploid nuclei. A key difference between meiosis and mitosis comes very early in the first prophase. Mitotic chromosomes are independent of one another. But in prophase 1 of meiosis, homologous chromosomes synapse. They pair up tightly. This diploid nucleus has 2 pairs of homologous chromosomes, n equals 2 and 2n equals 4. Note that the pairs have synapsed. During prophase 1, there is genetic exchange, crossing over between chromosomes. Chiasmata are visible evidence of crossing over. Prometaphase begins as the nuclear envelope disappears and the spindle invades the nuclear space. Pairs of homologous chromosomes move together to the equatorial plate for metaphase 1. Now comes the second key difference between mitosis and meiosis I. In anaphase of mitosis, the chromatids separate. In anaphase 1 of meiosis, the homologous chromosomes separate. The chromosomes moving to the poles in anaphase 1 consist of 2 chromatids each. This step reduces the chromosome number from diploid to haploid. Following telophase 1, there may be an interphase called interkinesis.
Conclusion. TUBB8 mutations have dominant negative effects that disrupt microtubule behavior and oocyte meiotic spindle assembly and maturation, causing female infertility.
There are 4 true stages of the cell cycle: G1, S, G2, and M. A 5th stage, G0, is where the cell remains indefinitely until it begins the cell cycle again. Some cells may never leave the G0 stage. Others that go through division rapidly, such as bone marrow cells, may skip the G0 stage entirely. Cells begin the cell cycle by entering interphase. In interphase, the cell prepares for division by duplicating DNA. Interphase includes the stages G1, S, and G2. In G1, the cell synthesizes DNA and protein and increases organelle and cytoskeletal elements. The chromatin consists of long, slender rods jumbled together in the nucleus. S is the synthesis stage. Nuclear DNA replicates along its centrioles, which are small structures made up mostly of microtubules that associate with the chromosome's DNA. The chromatins begin to coil so that they shorten and thicken. In G2, RNA and protein are synthesized. Proofreading also occurs. The DNA replicated in the S phase is checked for mistakes. If mistakes are found, the cell repairs them or receives a signal to self-destruct, called apoptosis. The difference between G2 and G1 is that in G1, DNA and protein are made, while in G2, RNA and protein are made. In the M stage, the cell divides into 2 identical cells through divisions of the nucleus, mitosis, and cytoplasm, cytokinesis. Over 5 different stages, the centrioles pull the chromosomes apart and the cell divides in 2. First, in prophase, the duplicated chromosomes appear and then split into identical halves called chromatids, attached by spindles called centromeres. The nuclear membrane disappears and spindle fibers form in the cytoplasm,